So there's your link for lecture 4. Make sure you guys have it watched before next class, okay? Now, it's going to take me a second because I have to open up all the stuff that I had to close out of. Okay, so today I'm going to do everything that I can to review parts of the cell as effectively as I can. But the thing about the lectures is you guys have to watch those lectures because I can't review everything because it's more important, honestly, that you guys stop and you work with this material on your own, on worksheets and in the games that we play and stuff like that. So I'm going to show you some videos that offer a lot of bang for the buck. And try to show you lots of cell parts in an animation working together to do a task for the cell. Now, gone are the days where you're going to have a simple multiple choice question that if you know like this cell part, you'll be able to get that multiple question right. Now, to answer AP questions over cell parts, you guys are going to have to know like several cell parts and how they interact. So there might be a question that you have to, in order to answer it correctly, you're going to have to know what 4 different cell structures do. And if there's one of those 4 you don't know, then you're going to be guessing on that question, okay? All right, I think I have that up. Go ahead and start digging out worksheet 3.3 for me because we're going to be using that and answering some questions as we go. 3.3. 3.3 looks like this. Okay. I am presenting. We're good to go. All right, guys. The best way is to try to use as many visuals as you possibly can because the majority of your AP questions are going to be about diagrams or they're going to be about tables and graphs and stuff like that. And I have no idea what these diagrams are going to look like. Sometimes they're really clear images, kind of like what I show you here. And you have to then, look at these images and answer questions about them. Sometimes, though, the images are so difficult to look at. Like this image, this was off the AP test, okay? So you had to be able to figure out like cell parts from looking at that image, okay? All right, so let's start with some animations here. Just note, you will eventually have to narrate one of these for me. And how you narrate it is you actually are going to type what it is showing you. Great, glad that's crashed. You're going to have to type what you are watching on the screen, okay? We might not get to it today, but we might open up with it next class to review, okay? All right, so one thing where I can teach you about a lot of different cell parts is the process of protein synthesis because there's several cell parts that have to work together. And by the way, if you don't make a protein correct, you're sick. You have a genetic disease. Sometimes if you don't make a protein correct, just one protein, you're not even born. That's how important it is that we make our proteins correctly. And several cell parts have to work together for that to happen, okay? So we're going to be zooming in on the nucleus here and on DNA. Now, you need to be able to tell me that DNA comes in multiple forms. Sometimes it coils up into chromosomes, but you can then uncoil those chromosomes. It really is just a string of DNA that is, we call it in chromatin form. Now, another piece of information you need to know is those proteins, those histones. DNA can wrap around those proteins, and that's what helps the DNA coil up. So if I gave you some string and I say wrap it up, most of you would wrap it around your finger, right? So those histones are like a structure, a stone that we can wrap that stringy DNA around to coil it up and to organize it. There'll be more on histones in a different unit, okay? Now, DNA codes for proteins. A gene is the code for one polypeptide. But if you remember, a lot of proteins aren't just one polypeptide big. They're 2 or they're 3 or they're 4. So here's an easy question that it's an easy one to miss. If you need to make hemoglobin, and hemoglobin is made of 4 polypeptides, guess how many genes are going to have to be read to make one molecule of hemoglobin? It's 4, because one gene is one polypeptide, okay? So here we have a gene. And a gene is just the instructions for building that polypeptide. Now, we do not want to send the instructions out to where the protein is actually made. So we're making a copy of it. So this enzyme right here is called RNA polymerase. It is making a RNA polymer, okay? So that molecule that's in the middle of the DNA there, that's nucleic acid. It's called RNA. It's made of RNA nucleotides. And then that mRNA is going to be able to leave and carry the instructions to a ribosome, okay? That ribosome is out in the nucleus. Now, we're going to learn more about this. This is called mRNA processing. That's coming in like unit 5, okay? So that mRNA, after it's made, it actually has to be worked on a little bit before a ribosome can read it, okay? So now that the mRNA can come out of the nucleus, it's in the cytoplasm, and here's our next cell part, a ribosome. You need to know more about ribosomes. Why? Because we can target ribosomes, make them stop working on like an infectious organism that's causing you an infection like malaria. And if we can target their ribosomes, then they can't make proteins and then that organism will die. Okay, so ribosomes are something that we can target. Some things that you need to know about ribosomes, and there's that are in addition to what you maybe learned in the past. There's 2 subunits to them. If you saw like 2 different subunits kind of came together and wrapped around this mRNA, where do ribosomes come from? I'll tell you what, take that worksheet, flip it over to that front page. If you're not there, let me show you something. You guys have been shown this all the time, but you maybe never really realized. So here's that nucleus, right? That's an easy one. That's the nucleus. Do you see that dark structure in the nucleus? Haven't you always noticed that on cell diagrams before? Like they always put this dark circle inside the nucleus. That's called the nucleolus, and that's where ribosomes come from. Okay, so ribosomes are made by an area, a structure inside the nucleus, which is called the nucleolus. Ribosomes are made of protein and rRNA. rRNA. R stands for ribosome. Okay, so ribosomal RNA is a nucleic acid, and that is built into the structure of a ribosome. Okay, so that's some additional new stuff that you guys need to make note of. Okay, all the job of the ribosome is to do is to get amino acids and put them in a chain to form the primary structure of a protein. Okay, we'll learn more about this process, but I just want to show you, this is why I always show amino acids as circles. And so all a protein is a chain of amino acids put together. The ribosome helps this take place, okay? All right, now, ribosomes, you're about ready to answer a question for me on chat. Ribosomes come in 2 forms, okay? So this ribosome, I want you to tell me what type of ribosome this is, okay? Watch what's going to happen. The ribosome is going to release that polypeptide. It's going to fold up into its tertiary structure. Now you know this, right? And now we have a protein that has been placed in the cytoplasm, okay? So let's go back to my question and everyone open up chat. This ribosome that we just watched, there's 2 types of ribosomes. I want you to name what type of ribosome that one is. Don't put enter yet, just type it into chat. No one push enter, and on 3, we will push enter together. Okay, what type of ribosome did you just watch? Okay, 1, 2, 3, send. Oh, I even said the wrong answer. Okay, so notice we have attached and we have unattached on our list, okay? So if a ribosome is just in the cytoplasm, it's called an unattached ribosome, or you can call it a free ribosome. If that ribosome is attached onto the rough ER, then you've got what's called an attached ribosome. So we didn't see any rough ER. This was an unattached or a free ribosome. They make proteins that are just going to be released in the cytoplasm, and the cell is going to keep those proteins and use them. They might be enzymes. They might go into the nucleus and be enzymes like we saw here at the beginning, that big blob that was copying. Hello, fast forward here. This big blob you're looking at here, that's RNA polymerase. That was an enzyme. So that was a protein that was made by an unattached ribosome. All right, let's go to that worksheet real quick. Let's go to number 3. In lecture 4, I will teach you all about kinases. They're involved in signal transduction pathways. And you, at this point, can know nothing about a kinase, but as soon as I tell you on number 3 that the kinase is an enzyme that's in the cytoplasm, was this kinase made by a free or an attached ribosome, you should be able to tell me that answer. any protein that is in the cell, in the cytoplasm, or in the nucleus, that was made by a ribosome, a free ribosome. Okay, I'm going to show you what those attached ribosomes are going to do in this next part. Okay, all right, so let's take a look at what we mean by an attached ribosome. So, all right, so this next one, we're gonna start already. That mRNA is made. That mRNA, that little red string, mRNA is coming out of the nucleus, and it's gonna go to a ribosome, but this is called an attached ribosome because it attaches onto the rough ER. So that black spot right there, that is a ribosome. They're super small, and they can attach onto the surface of the rough ER. They're still gonna do the same thing. They're gonna make a protein, but they're gonna put that protein inside the rough ER, right there, that blue thing. That's a protein. Some proteins are in the lumen, and look at those green things. Those are proteins. They're just embedded in the membrane of the rough ER, okay? So the rough ER is the modifier. It can attach stuff to the proteins, and when it's done, it's gonna send it to the Golgi. Now, I've got two different proteins you can see here. I got the green ones that are embedded in a membrane of the Golgi, and then I have the blue ones, which are in the inside, which is called the lumen of the Golgi. The Golgi is the post office. It can tag proteins to go 3 places and 3 places only, and you need to memorize this, okay? Sometimes it's going to tag those proteins to go to the cell membrane. So watch what happens to those 2 types. Notice the green ones got embedded. So in the 4th lecture, when you're learning about transport proteins, receptor proteins that are embedded in the phospholipid bilayer of the membrane, you know how they got there. The Golgi sent them in a vesicle. Okay, the blue ones, those are proteins that cells make and release. So can you give me an example of a molecule that, or protein that a cell makes and then releases out of the cell into the bloodstream? Everyone give me a guess in that chat. Can you name me a protein that this blue one might be? So that is a protein that got made by a cell and got released from that cell. And now that protein is going to travel to a different part of the body. See a couple answers there. Okay. Now the receptor would be the green one. Receptor proteins are stuck in the membrane of cells. Nice, Vince. Thank you. Estrogen is a hormone, but that one, I'm glad you asked us. We're going to answer a question about it. Estrogen is a steroid. A steroid is a fat, not a protein. Can anyone tell me what cell part makes fats? Because that cell part would have made the estrogen. Awesome. Thank you, Michaela. So to help you remember this, when we need our hair to be smooth and our skin to be smooth, we put fat all over it. That's all conditioner is. That's all body lotion is. It's a bunch of fat. And you rub that on your skin and you rub it in your hair. So to make things smooth. So you might remember smooth ER, fats. We use fats to make things smooth. That's how you can link those 2 together. So estrogen would have been made by the smooth ER. Okay. Let's see if I actually have that question somewhere here that we can go ahead and if I am, perfect. 13A. 13A. You should be able to answer, oops, hit the wrong thing. You should be able to answer that question now. Cells that produce and release a large amount of steroids like estrogen would require more of the part that actually makes the steroid, which would be smooth ER. Okay. All right. Why we're here. Cells that line the trachea and produce and secrete mucin, which I tell you it's a protein, but on the AP test, they're not going to tell you a protein. But how can you tell that mucin is a protein if they don't tell you that? Because proteins are named with what at the end? IN. So that's a little fact that you have to have memorized. And if you don't have that one memorized, you're messed up on a lot of questions because they're expecting you to know that we name proteins with IN on the end. So if they're talking about a protein you never heard of, like mucin, you still know, oh, they're dealing with the protein. This is protein production question, okay? So to make a protein and release it like mucin, like adrenaline, like insulin, okay? So going back to that, that one that I just showed you right here, that blue thing, that is a protein hormone. That could be adrenaline. That could be estrogen. That could be oxytocin. That could be prolactin, okay? One cell makes it and then dumps it out. And then that molecule will travel to a different cell in the body, okay? So on this one right here, any cell, if its job is to make protein and kick it out of the cell, it needs a ton of Golgi to tag it to send it to the cell membrane, okay? Speaking of, so we're talking about specialized cells here on number 13. If I say list a specialized cell, just name any cell in the body, muscle cells, skin cells, melanocytes, white blood cells, red blood cells. Those are specialized cells. And they have features that are going to be different than other cells that have a completely different job, okay? Like a red blood cell doesn't even have a nucleus. Why? Because then there's more room for hemoglobin. So it doesn't need a nucleus because that cell is going to be gone and destroyed by your liver in about 30 days, okay? So it doesn't even need a nucleus. It's not going to live very long, okay? What you're looking at now is two specialized cells, okay? Notice they look different. These look like they're having a bad hair day. This looks like some of my 8 a.m. students. When Gage, he always got the highest hair in the morning. Cracks me up. Okay. And this other cell that you're seeing here, that's a different type of cell. It's called a goblet cell. It doesn't have that hair-like extension, which is cilia, by the way. Okay. So you should be able to tell me, basically why these two cells have different structures and what those structures are going to help them do. Okay. We're going to go ahead and zoom in on specifically. We're going to zoom in on this goblet cell. The job of this goblet cell is to secrete protein. And we're looking at cells actually lining your airway. If I can back it up a little bit, this is all about cells. So we're going to see cells here. Come on, you can play for me. That are lining the airway of your lungs, okay? So your airway is just coated with mucus. Mucus is mostly a protein called mucin. And the ciliated cells help that mucus flow and come out of your lungs so you don't get infections when you breathe in bacteria and stuff like that. So that's kind of what it's showing you here is your airway is just coated with mucus. Where does that mucus come from? By the way, now you know why they named it Mucinex. Mucin, that's the protein that's in your mucus. Mucinex breaks it up so that that mucus can come out of your chest so you're not going to get like pneumonia or something, okay? All right. So we're going to zoom in on this one type of cell. We're going to take a look at this goblet cell, okay? Now, look at that image. You see those circles? I have no idea what kind of image they're going to show you on an AP exam, but this right here looks really similar to, hold on, let me get rid of this stuff. Let's go to this question on worksheet 3. They're showing you almost the exact same thing that I was showing you in that movie, okay? All right, so let's watch the movie, and then we're going to come back and we're going to answer this diagram, all right? Okay, so this whole movie that we're going to watch here is about how this protein gets made and released from the goblet cells. This is one protein. That's the mucin, okay? Notice it's really branched. So you can see what Mucinex does. Mucinex comes in and actually will break down that protein so that it's not getting caught inside your lungs, and it can then, the pieces of it can move out a little bit easier, okay? All right, so we've got to make a protein. The instructions for mucin is in the DNA. This enzyme is reading the DNA and making mRNA, all right? Making mRNA. By the way, everyone open up chat for me real quick. In chat, I want you to name, but don't hit submit until I count down. What are those purple things? What are these purple things flying in on this image? You have to be able to look at images like this and know what is going on your AP test, okay? All right, so what are those purple things that are flying in there? Put an answer down in chat. All right, 3, 2, 1, submit. Okay. What are the monomers of nucleic acids? Nucleotides. If you need to build a nucleic acid like mRNA, you need a bunch of nucleotides. Those are mRNA nucleotides. Okay, so they're being strung together into mRNA. And now that the mRNA is made, it's a copy of the instructions, out it comes, okay? Now watch, here comes a ribosome. We're gonna see all kinds of new stuff happening right now, okay? Again, ribosome, 2 subunits. Ribosomes are made by the nucleolus. Ribosomes are made of rRNA and protein. So they're gonna come together onto mRNA, but watch what's gonna happen. Something different than what I showed you in the past, okay? The ribosomes are going to start to make a protein, but whoa, what is this big blob thing that just attached on? And what's happening to my ribosomes? Bam. We just had a lunar landing. That's what it looks like. It looks like a spaceship just landed on the surface of the moon. Here's some craters, right? That ain't the moon. But here's what I want you to do in chat. I want you to tell me, what did this ribosome hook onto? What is that surface that we're looking at there? Don't hit enter until I say go. What did that ribosome just land on? Should be a short type in because it's 3 letters long if you want to abbreviate it. All right, hit submit on 3, 2, 1, go. Submit it. Yeah. I have to tell you, first block, the first person who posted something was wrong, and everyone put that exact same wrong answer all the way down. And he said that was the plasma membrane. That's not the plasma membrane. That's the rough ER, because that's the only thing that ribosomes can hook onto is the rough ER, okay? Now, this ribosome is now in the rough ER. It's still going to make a protein, but it's going to put that protein, which is that little brown part there. They need to change the color of that. That looks awful. Okay, so it's making the protein, and it's putting the protein inside the rough ER. So there goes the mucin, okay? And it's showing you now these ribosomes, they put the mucin in here, but now these vesicles that contain the mucin are coming off of the rough ER, and they're making their way to the Golgi, okay? Now you're inside the Golgi. The mucin is inside the Golgi, and it's getting modified, and the Golgi puts it in a vesicle and then releases that vesicle out into the cytoplasm. Cytoskeleton, we haven't talked about it yet. Do you see the green things? Do you see the big tube things? Those are different types of cytoskeleton. And cytoskeleton can be used as highways in which these vesicles are walked. They just don't float throughout the cytoplasm. They're actually walked by motor proteins that can walk along the cytoskeleton. And it does. It looks like they have 2 legs, and they just walk along the cytoskeleton. Eventually, that vesicle makes its way to the membrane. It fuses with the membrane, and out comes that mucin, okay? All right, speaking of, here's what I mean by fine details and why you have to watch those lectures and pay attention, okay? So ×2 speed means that you're probably have to go back and watch it again. You have to know minor details, like, okay, cytoskeleton, what else is cytoskeleton used for? Well, let's go back to the cilia here, okay? So we're about ready to go back in, and we're gonna take a look at cells here that have cilia on them. I was hoping for another image, okay? The cilia has cytoskeleton in it. And cilia can't move unless there's cytoskeleton and motor proteins working together to make the cilia do that, okay? Why do we expect you to know something that's simple like that? Because there are protists that can make you sick, and they have cilia. So if you can damage their cytoskeleton, they can't move their cilia, they can't move, and that's a way to kill them, okay? So again, we learn about all these parts so we can figure out how to exploit, organisms, target them, and kill them when we don't want them in you or in a certain area, whatever, okay? All right, let's go to this worksheet real quick, okay? You just watch that process happen, a mucin being made and being secreted from a cell. Let's see if some of this makes sense, okay? And then a few more things that we need to add here that are important. It says, microscopic images taking over time of ovalbumin production in chick oviduct cells. I'm not going to teach you any of this stuff, but what I have taught you is this. If a molecule ends in IN, you're dealing with protein production. That's what this whole question is about. Move this out of the way. Okay, now it says ovalbumin was detected using a fluorescent dye. Fluorescent means it fluoresces, it lights up. So the reason why we tag molecules with a dye is so we can watch where they go, so we can learn about them, okay? I want you to circle for me and write something. Circle fluorescent dye. Fluorescent dye. We're going to go ahead and take care of this now, and I don't have to wait till molecular genetics to teach you this, okay? Lots of times those dyes are radioactive. And you guys hear the word radioactive, and you've watched too many movies, and you think that if we use something that's radioactive on a person, that they're going to turn into something like Spider-Man or the Hulk, and they're going to have these, mutated DNA. I need you to understand that sometimes things like dyes are radioactive. And we wouldn't be able to watch where these molecules go unless they were radioactive. And we have devices that can see where radioactive material goes. So I want you to know that radioactive doesn't mean bad. It doesn't always mean cancer, okay? We can use substances that are radioactive in medicine. If you are bleeding internally, that means that your abdomen is filling up with blood. And if we don't figure out where the hole is and how to plug it so you stop bleeding out, you are about ready to die. So one thing that we can do is we can inject radioactive isotopes into your bloodstream. And they're going to move, and some of them are going to shoot out that hole that you have in your circulatory system. And we now know where you are bleeding out. So now we know where we need to go in and stop the bleeding to save your life, okay? So just note, when you see radioactive isotopes, we're using them to track and trace molecules, and it's a good thing. We're not giving you cancer by using them, okay? All right, let's see if these images make sense to you now. Can you tell where the nucleus is? Is the protein that's being made ever in the nucleus? No, it was in the cytoplasm because the ribosomes aren't in the nucleus, they're in the cytoplasm. So notice the nucleus never lights up because that protein that lights up is never in the nucleus, right? Okay, one of the first things that lights up right around that nucleus, that would be the rough ER. So we have the protein is now in the rough ER, okay? Now what's lighting up? Something a little bit more here. That would be the Golgi is starting to light up as it's getting that protein. Now we have all of these little circles. Those would be vesicles that have the protein in them. And they are making their way now to this cell membrane. And now that light up is happening outside because that ovalbumin has been dumped out of the cell. And we can see that happen if we tag that molecule. And we can watch where it goes, okay? So that was an image that was on the AP exam. And it all boils down to, do you remember protein production and the parts that take place, or the parts that are involved in making that protein, okay? All right, so speaking of, let's answer number 11. You can go in and answer more of it. Let's just list the cell parts, okay? So I covered things like the nucleus is involved in protein production. Attached ribosomes. Rough ER. Vesicles. Cytoskeleton, because the vesicles have to be walked along the cytoskeleton. Golgi. Plasma membrane. Okay, so we just covered a lot of cell parts there really quickly. One last thing that I want to show you, because I didn't finish this video, okay? So I mentioned that the Golgi can tag proteins for 3 different destinations, okay? This is a really fast video. This is going to show you everything in like 8 seconds, all right? So watch the screen. Here we go. 3 different proteins sent to the Golgi. Golgi can tag them for 3 different destinations, okay? I already kind of mentioned this one to you. That would be like a protein hormone that got released. That could be insulin, and this is a beta cell that just made it. That could be a receptor protein. It's stuck in the cell membrane. It's not going anywhere. But let's take a look at this. What is that? Okay. So the last part of this video I need to show you, okay, is that when cells engulf material, and some cells do, some cells don't. So let's say this is a white blood cell, and it has to swallow up that foreign particle that's in your body and destroy it. The destroyers are the lysosomes. Lysosomes contain enzymes that can break down molecules. Those proteins came from the Golgi in the same process. So some of these proteins that were made by those attached ribosomes can ultimately be tagged to become proteins in a lysosome, okay? Also, note everything that just made a vesicle for me. I'm going to back this up a little bit. All of these parts are part of the endomembrane system. If something can make a vesicle and ship it to something else, then you say that that cell part is part of the endomembrane system, okay? So note, ribosomes don't make vesicles. They just make proteins. They're not part of the endomembrane system, okay? The first thing that we really mention is the rough ER, but also note, this is the nucleus. Do you see how the rough ER membrane is also like attached to the nuclear membrane? So the nucleus is part of the endomembrane system because the membrane around that nucleus is actually just leads right into the membrane of the rough ER, okay? Rough ER can make a vesicle. It's part of the endomembrane system, okay? The Golgi can receive vesicles. It can send out vesicles. So it's part of the endomembrane system. Let's fast forward a little bit here, okay? The plasma membrane can receive vesicles. So it's part of the endomembrane system. They can exchange substances with vesicles because they're all made of a phospholipid bilayer, which links back to that theory about how all these cell parts became to be by infoldings of the membrane, okay? And then lysosomes, part of the endomembrane system because they send and receive vesicles, okay? So is the smooth ER. But in this video, did I show you a mitochondria sending out a vesicle? No, they can't because they have two membranes. They can't form vesicles. Same with the chloroplast. They have two membranes. They cannot form vesicles. They cannot send and receive vesicles because their outermost structures look different than the other members of the endomembrane system, which brings me to number 6 on your worksheet. Let's answer that real quick, and then we're going to pause and do something else. Number 6, right there. Which cell parts are part of the endomembrane system? Let's answer number 6 real quick. So anything that is a single phospholipid bilayer that can send and receive molecules using vesicles is a member of the endomembrane system. It's like an exclusive club. You have to be able to do something to be in the endomembrane club. Endo means inside, membrane means they are inside structures that are made of a membrane, and they can send and receive vesicles. Okay? All right, I cruised through a lot of information. Let's see what you remember. We're going to play a GEMKIT real quick. That's going to review stuff from the first 3. Vincent, try not to get voted out in the first 2 minutes. Okay, so on the back of the worksheet, let's take a look at some of the ones on 13 here. All right, so let's do B. That should be an easy one. So we saw goblet cells make the mucin and secrete it. What would be the structures that are on those other cells that move the mucus? And those are like short hair-like extensions. And so if something's on the top of it, it gets moved. Mm-hmm. Yep. Okay, D is kind of tricky. It says cells that contain a lot of protein. So that means the protein stays inside of them. If it contains a lot of proteins, they're not shipping proteins out. They're making proteins and it's staying inside of them. So what would they have a lot of to be able to meet those high protein demands, like in muscle cells?
Nuclei.
Ah, that's a great point. So actually that's true, is muscle cells actually have multiple nuclei so that transcription can happen enough to meet the protein demands. So if you put nuclei, I love it. That's thinking outside the box. You could also put unattached ribosomes. So there'd be ribosomes all clustered around all those nuclei to churn out the actin and the myosin proteins needed so that a muscle cell can contract. E, cells that ingest and digest foreign substances. Okay, so I showed you that video and I said, here I have a cell, and let's just assume it's a certain type of cell, and it takes in molecules.
Are released into the extracellular space. Soluble proteins on the outside.
All right, so here we go. So this cell just took in something and is going to digest it. It would need a lot of lysosomes if it needs to bring in particles and digest them. Okay. In the notes, it talks about specialized cells. We talked about sperm cells. So cells like sperm cells, they will need a lot of ATP to power what? What's that little flippy thing called? It's a tail, but it's called a flagella. And note that they don't have any other organelles. Sperm cells are tiny, and big things don't move far. And that's what a sperm cell has to do, is it's swimming a marathon. So it doesn't need rough ER. It doesn't need smooth ER. It doesn't need any of those things because it's about ready to die in about 3 days, if not before, because a female's white blood cells will take those suckers out. All right, 4 minutes. Let's talk about number 12. Identify a feature found in both of these cells that increases surface area so these cells can exchange nutrients with the environment more efficiently. Have you ever wondered why red blood cells have the shape that they have? Why is there an indent in the middle? How come your red blood cells aren't just like perfect little circles floating around? Well, that indent actually then increases surface area. That's a fold. So red blood cells that are folded would give an organism an advantage because then they can take in and release oxygen through their surface area, and they can do it better because they have more surface area because their plasma membrane is folded. Okay, so any cell that needs to like absorb or release a lot of things, lots of times we see folded membranes so that they have the surface area where those things can go in and out of the cell. Man, we cruised through that. Let's go back to number 7. Let's see if you remember some stuff from lecture 1, number 7 on the first page. Can you name me 3 cell parts in the diagram that contain DNA? I'll give you the easy one, the nucleus. What are the other 2? And it's evidence that their ancestors were once bacteria. Anybody? I'm not going to give you this one. It's evidence that their ancestors were once free-living bacteria. So which cell parts do we believe originated from bacteria? Oh, I'm sorry. I'm yelling at you all to answer, and my volume was down because of that video. So I'm sorry. What'd you guys say?
Chloroplast and mitochondria.
Thank you very much. All right, we got 2 minutes, so we can go ahead and start exiting out. Just note, you guys need to watch lecture 4, or you and your partner will be absolutely clueless when it comes to building your models of a signal transduction pathway that starts with a G protein linked receptor. Okay? All right, we'll see you guys.